All right, let's come back. Continue to talk about epigenetic regulation. So again, coming back to this central dogma of molecular biology, we got the DNA, and this is transcription part, and become RNA processing, and become protein. This is translation part. The epigenetics part is actually from the DNA to RNA part. It controls the kind of helping to affecting the transcriptional regulation. And the two major forms of epigenetics, we know that it includes histone modification and the DNA methylation, these two parts. And the first one is histone modification. Just to kick in a little bit biology about that. We know that this is how DNA is organized, okay? And this is really the chromosome and the entire chromosome, but if you really zoom in one part of it, actually it wraps in this way. And then if you further zoom in, this is a chromatin, and actually there's 4 different histone markers, 4 different histones, and then the DNA double strand wrap around it, and of course this is the double helix part of it. So you can see this is how the chromosome is organized. And this is the same thing from just a different figure and the chromosome level all the way to double helix. So you can see this is double helix DNA, and then this wrapping around the histones, and it becomes a chromosome chromatin, and further goes up. Once we zoom into one nucleosome, and you will see that actually there's 4 different histones, and histone 2A, 2B dimers, and histone 3 and histone 4. And you can see that these are the 4 different histones, and the double helix DNA wrapping around it. And each nucleosome is about 146 to 147 nucleotides, and in terms of the DNA. Okay, so this part you can see this histone part, and is very good, just the solid protein structures, and the DNA is wrapping around it, right? We know this. But what really happens is this part. So you can see that each individual histone, and you will see there's a small tail that is stitching out, and this is something that really receiving biological signals. Okay, so let's coming back to this type of view. So you got one histone. This is histone 3, and this is the solid protein structure part, and this is the histone tail, and this is kind of disordered and an unstructured part of it. But once you got this, and you should be able to. This is also an amino acid sequences. So there's many different amino acid sequences, and at different locations they are different. And what happens is at different locations there are some different type of modifications that can really occur. Okay, and what the modifications include this part, include acetylation, phosphorylation, methylation, and ubiquination, and with different type of flags. As you can see, there's different flags that correspond to different type of modification. And with different type of combinations, and this will give you a different biological meanings. So one of the example, if we talk about the histone 3, K4 dimethylation, the histone marker we talked about in the earlier stage, that really means that histone 3, K4 is the 4th position in the tail, which is the lysine, and has 2 methylation sites on that. So you will have those type of coding, and this is also called histone code. All right. So computational methods for identifying chromatin signatures, and this will be just very brief because, as I mentioned, there's not too many methodologies, at least I can find out. All right. So this is an earlier slide that tells you the classes of ChIP6 signals. This is actually the 3rd type. So there's no peaks, and there's not a peak and follow up with transcripts. Rather, it's just a broader range that has an enriched signal comparing to the background signals. And this will be what we are trying to identify. The way to do that, methodology-wise, this is a paper I think is published in NAR, Nucleic Acid Research, and in 2009. This is a paper published by ChIP group. So remember that Cell paper, 2007 Cell paper on 20-something histone markers. And this is the paper they really, the group that work on that. So that's why I think, because there's not really too many methodologies has been developed. And that's number one. Number two is there's really not too much benchmark data can help us to evaluate which methodology is doing better than others. So I guess that group is at least credible. And to do that, for their methodologies, they will include two different stages. One is to identify the candidate regions. And what the factors they are using is they first will break the chromosome into different pieces. Again, this is a bin approach. I think they were using about 200 base pair as their bin size. Okay? And for each individual bin, and they have a minimum threshold as a background. So this is for this part, you don't really need a control sample. They just calculate what's the minimum amount to qualify to be an enriched region, the number of rays in each individual bin. And here is actually how they decide this minimum threshold. And this is based on a Poisson model, and this is the p-value that you are targeting. Again, this is just understand the concept. That will be fun. There's no way that we need to write a program to do that. They actually have the program. It's available. And using this type of equation, that can help you to decide the minimum threshold for background. For example, for this case, they are using is a 2 as a background. So if there's for each bin, there's less than 2 fragments, and they will consider it's a background region. And as you can see here, that for this one, most of the bins in this region will pass this threshold. So these are really the enriched regions, but there are a couple of gaps. For example, one of the gaps is here, right? So at that gap, in that particular bin, there's no signals at all, or the signal is less than 2, right? And so they define this as gaps. And the next step is they will combine the signals of different bins together and using a much more complicated statistical model and trying to see if I see this many consecutive bins that has the signal with a few gaps in the middle, what is the genome-wide probability that this is a background or not? Does that make sense? So in the first stage, they divided the genome into different bins. And for each individual bin, just count the numbers of fragments fall into that. And if there's above certain threshold, they will consider that is a good bin, right? This is the enriched bin. But once you look for different bins together, and this one is okay, but there's a gap. And suddenly there's a gap, and there's another bin is okay, and another gap, and 2 bins are okay. So in random, even though this is the enriched region, you potentially can see this, right? Because some part you just didn't sample enough, and some part you just didn't detect the signals. And and this is the first step. And the second step is based on this type of patterns, there's so many different bins that has a signal, and suddenly there's 1 or 2 gaps. And what is the probability that this is one enriched island? And this will be the way that the concept they do. So the first one is to identify the candidate regions. The second one is to identify the enriched islands. And this approach can be used with and without controls. So that is the way that they design that. The paper is here. It's published in bioinformatics, and I think they also have the code available for you to use. So I just want to have a little bit side notes on that. And for this, this is, as I mentioned several times, this is the only approach I was able to find. And there is another publication using hidden Markov model. And the same, looks like they are targeting a same problem. But what they are trying to identify is the differences between two samples, differential chromatin states. And that will be different. But for our purpose, this is more equivalent to the peak binding part in the transcription factor binding sites. And this is look for the broader enriched region in the histone markers. Okay? That make sense? Question? All right. Let's move on. I know there will be no questions. All right. And there is another piece of a computational part that I want to talk about regarding the functions, regarding the histone markers, which is the functions of histone marks. So this is a paper published in 2010, last year, and in Nature. So what they did is, based on 38 different histone methylation and acetylation markers, and for different histone variants and RNA polymerase and other factors in these T cells. And the goal of this study is, we can always say, okay, let's do the ChIP-seq data on this, let's say, 38 different histone markers. And histone 3K4 dimethylation is enriched in promoter regions, in enhancer regions, or so on and so forth, right? But if we do that type of analysis, we have to know what the histone code really means. We have to know that histone 3K4 trimethylation is in the promoter. Histone 3K36 trimethylation is a transcript marker. We have to know all these things. But for most of the histone markers, we don't know what they really mean. Okay, so what is but we do have a lot of ChIP-seq has been done, and how we can learn that which histone mark or a combination of different histone marks in a region, what that encodes for. So this is the basic goal of this study, is to identify the functions of histone marks. And what they did is they called de novo discovery of chromatin states. And they used something called hidden Markov model, which I'm not going to go into that. And but the point here is it's a probabilistic model that model the multiple observed input. For us, here is the signals of various different histone marks. So we have 38 different histone marks, and this is observed input and generated by unobserved hidden state. This is how the model is organized. And these hidden states are actually the chromatin states. For example, which state is a promoter, which state is the enhancer, and so on and so forth. Let me directly jump into the results. So this is what they derived, published last year in Nature. I know it's small, but I really don't know how to make it big. So bear with me. So you can see these are all the histone marks they did. And this is one gene. It's in the promoter region, different exon region, and the intronic regions. And here is for different histone marks and for different bins in those regions, what the signal enrichment look like. Okay? So this is completely based on the experimental approach, nothing computational at this stage for this. Okay? And this is really not the best color combination, but there's no way I can change that. And here is a different state, the chromatin state. And I think, let me see. They are saying, okay, here it is. This is, these red ones, those are the promoter states. And so you can see that at this promoter region, based on the combination of these signals, and this is all the different promoter states. And this is, the blue ones are the transcript states. Let me see others. And the yellow ones are the active intergenic regions. So these are different chromatin states. And this is another figure that shows you here is 38, not really quite 38, but many different histone markers, what their signal intensities are. And here are different states. So this is the promoter states, and this is the combination of the histone markers in those regions. And this is the transcript states, active. I can really not see that. Intergenic states and some others. So if you're interested, go to this paper and to look into that. So the point here is not really putting there and trying to just confuse you. And this is not something that we need to do in the future either. So once we got those histone markers, okay, and ChIP-seq data, and we can use the previous approach to identify the enriched regions, that's one thing. And the second one is if there's more and more others showed up, the different histone markers, and most of them we really don't understand the biological functions of that. And this is one of the effort is trying to using computational tools based on our existing knowledge and to understand what is the functions of histone markers. Okay? If you do not understand the methodology and you don't understand, you cannot see the results. But I want to confirm, do you know what we are trying to do here for this one? So the first approach is the approach that we say is for one histone markers, we did ChIP-seq, we identify enriched region, and that one slide is showing that, right? So let me go back to this. This one is trying to identify enriched region for one histone mark. Once you do the experiment, you see which part of the chromosome is enriched. So this is what this approach is for. Okay, this is more useful for you. Once you got the new data, you just try their algorithm, and then you identify regions. This is the enrichment. And the later one that I talk about here is some computational effort based on different histone markers, trying to understand what histone marker encode for what. Okay, we know histone 3 K-fold trimethylation encode for promoter, but there's many histone markers we don't understand what their biological meanings are, and this is one of the ways that help you that. All right. If you still don't understand, go back to listen to your podcast, and hopefully that all the information I want to deliver is recorded in there. Are we okay? Are we okay? Okay. So next, I'm going to jump to the DNA methylation part, so DNA methylation, and actually it's also called CPG isolation, and the cytosine phosphate guanine, so CPG. And so what really happens is once you got this CPG dinucleotides, and the C of the CPG dinucleotides can be potentially methylated. Methylation means there's a CH3 groups that is modified on top of it. Okay, and it is strongly represented in repetitive sequence associated with retrovirus-derived sequences. This is based on the biological observation. It can be methylated and to generate this, they call it a 5-methylcytosine. So basically, it's a C and with the methylation groups on top of it. And but this is the interesting part of it. So if you don't do anything, sometimes it can be spontaneously deaminated to form thymine, meaning that methylated C can be changed to T if you don't really control, the cell doesn't really control well. Okay, it's easier to be changed to T. And this change is poorly recognized by DNA repair systems. Okay, so you cannot, the cell doesn't really have a good way to correct this change. And this is caused, so that's why that CPG to CG to TG mutation will be propagated. So there you will see this part cannot be really corrected. And the CPG levels are less frequent than predicted 1/16. That's the result of that. So we know that we have 4 letters. Let's say each one is 25% ACGT, right? And every dinucleotide, you should be able to, every 14th dinucleotide, you should be able to see 1 CpG if everything is just random. However, this in human genome, I don't know whether you did that analysis before. You probably just heard about the results. Actually, when I was a graduate student, and that is the first thing I look at. And I studied promoter regions a lot, and I did calculate the percentage of CpG that in the promoter regions, and also in some random selected regions. I think it's less than 4% of the dinucleotides are CpG at the time that I made the calculation. But it's much less than 1/16. And the reason for that is this CpG, the C, can be modified. And then if it is modified, it can be changed to TG. So we see a little bit more TG in the human genome, but the CpG frequency is much, much lower. Okay? And this may contribute to relative inactivity of a retro element. So we'll not talk about that. So I will have one slide to talk about why the CpG island is generated and in terms of evolution. But before that, let me talk a little bit about the DNA methylation-mediated repression. Why biologically, functionally, DNA methylation is important. So you can see that this is one gene here, okay? And this is the transcription starting site. This is the promoter region. And this is the CH3 group, which is the methylation group, is there. So once it's methylated, and it should have a transcription factor binds in this region. If there's a methylation, it's not there. But once it is there, and the transcription factors cannot really bind here. And therefore, the gene expression will be killed. And this is something that you want to keep in mind if you study the DNA methylation eventually. And most likely, once you go back to the promoter regions, if you identify, oh, this is the site that has been methylated or not, that affected the biological function, you want to go back to these regions to see which transcription factor potential binds in that region, and based on just the motif finding algorithms. But this is the first approach, why the DNA methylation can affect the gene expression. The second one is the CH3 group. There's a methylation binding protein that are just binding on this CH3. So the MDP is that there's some protein that is will be just like the methylated C, and it will bind there. So that will be generate the bigger protein. So the transcription factors and other protein complex cannot bind here. Therefore, gene expression level cannot be really occurred. And this is another type. So you can see that this is the DNTP, DNA methylation methyltransferase, and let me see what happens here. So you can see this transcription factors and these methylated transcription factors cannot bind here, and this DNTP actually recruited some of the acetylation factors, and this is acetylated. So this methylation not only affected on the transcription factor binding, but also affected the local chromatin structures. Okay, which means that in this part there's maybe some histone acetylation happens, and we know histone acetylation that mostly encodes for the active markers. But once this acetylation has been removed because of this methylation occurred here, and it will further kill the gene expression. So this is the third mechanism why DNA methylation can cause the repression. And this is the last one. Actually, you can see that if methylation happens in the gene, not in the promoter region, this is in the gene region. And if this methylation happens, and there's a methylation binding protein was attracted here, and it will actually affect the RNA polymerase II further going through this region. And that is another mechanism that this methylation can stop the transcription. Okay? So methylation in the promoter region and in the gene region really can help us, can affect the gene expression. And in terms of DNA methylation and the disease, so there's this figure actually shows you two different models. The first one is you can see that this part is supposed to be hypermethylated. Hypermethylation meaning the methylation signal is very dense in that region, so that this gene shouldn't really transcribe a lot. Okay, but once something happened in the cell, and this part is hypomethylated, meaning the methylation signal has been removed, and this gene that doesn't supposed to be there start to transcribe. That, if this is the oncogene, and that potentially will cause the cancer. Okay, and for this scenario, you can see there. I mean, they have the real example for all of those. For this scenario, you got a tumor suppressor gene here. And we can see this is hypomethylated. So the hypomethylation, you can see this is a methylation site. If this is a circle, an empty circle, that is not being methylated. It's a potential methylation site, but not being methylated. If there's a solid circle, that means it has been methylated. For this one, you can see this is not methylated. So this tumor suppressor gene will continue to express in a normal condition. But once people are sick, and this part is hypermethylated, and this tumor suppressor gene will be killed, and that is another mechanism that can cause cancer. And both these two mechanisms that we have been observing that in the cancer cells. This slide tells you the evolution of the CpG island. So why CpG island really occurred. Also, what is the CpG island? As I mentioned, that in the human genome, the CpG dinucleotides are rare. Okay, it's much more rare than it should be. By random, it's 1/16 dinucleotides there will be CpG, but we don't see as many in the human genome. And this is really because of the methylated C can become a T. But the only exception is in the promoter regions. In the promoter regions, we still see a huge amount of CpG dinucleotides. And the reason for that is because of evolution selection power. So you can see that this is the CpG dinucleotides, and this is the gene region. And these are intergenic region, means they are not very important. Okay? And this part is high methylation, this is high methylation, and this part is low methylation. All right? So what happens is once these methylated ones, methylated Cs, will be mutated to TG. CG becomes a TG. And if it's on the reverse complementary strand, it becomes CA, right? And if this really happened here, and all these things will be changed to other nucleotides. So that's why that CpG it's become very rare. However, in the promoter region here, if there is a CpG has been methylated or heavily methylated, if we also change to TG, and because the promoter region is kind of important, okay, and if it's changed, and that individual will be killed. It's not going to survive, right? So because of this selection power, evolution selection power, we see a great deal of CpG dinucleotides in the promoter region comparing to any other region that are not important. Does that make sense? And which also tells us that in the promoter regions, the methylation signals are usually low. Okay? Because if it is high, and the CpG will be changed to TG, and eventually the CG doesn't occur anymore. Okay? So this is how the CpG island is generated. It's just because of the mediation of the methylation. And next topic is what is the experimental way to study the DNA methylation? And the first approach is very similar to ChIP-seq. Actually, I haven't tried, but I think the enriched region in the chromatin markers that we talk about to identify those technologies can be used here as well. So I'm not going to go over this again, but you can just think this protein of interest is actually the CH3 group, the methylation group. And once you do this experiment, you can use 3 different ways. The first one is you can use methylation DNA immunoprecipitation if DNA containing methylated C using a antibody, monoclonal antibody. So you can just use an antibody to target on the methylation binding protein. And that is one way to do it. So because a lot of methylation sites, they will be attracting this methylation binding protein there. If you use the antibody to target this methylation binding protein, you will be able to identify the methylation site. And the second one is to use MDBD affinity purification. So basically what happens is you create a column that has these methylation binding proteins in there. And then you pass your DNA through that column. The DNA that are methylated will be trapped in the column and went through. Hopefully they don't have too many methylation left. So this is 2 different type of thing. And the third type is a kind of pretty rare. It's use a CXXC affinity purification assay. It's called ChIP assay. But the point here is these are the immunoprecipitated DNA containing unmethylated CpG using this. So instead of you passing a column, only the methylated part has been trapped there. Actually, only the unmethylated part will be trapped there. So everything flows through actually was methylated. Okay? But anyways, regardless of which approach you use here, and the methylation, you are using this kind of antibody affinity purification based method. And it's very much similar to the ChIP-seq type of technology. The second technology, which becomes more common to use, is called bisulfite sequencing treated. And so the biochemistry base for this is bisulfite treatment will change the C to U, but leave the methylated C unchanged. Okay, so it's different from methylated C become T in the random situation. So the bisulfite treatment can change all the C to U, except for the methylated C. So what happens is once you got the cell, you are going to treat the cell. So this is the original DNA. So you can see there's still a lot of CGs in here. Some of them will be methylated, others will not be methylated, and things like that. After you do the bisulfite treatment, and this methylated C will be captured here. So you can see this C is still here. But all the remaining unmethylated C will become a U. And this one, although there's a CG here, this C is not methylated, so it will also be changed to U, and so on and so forth. So what you are going to do is you got the cell, you treat with bisulfite and to do the conversion, and then you sequence both treated and untreated cells. And once you're sequencing these 2 different conditions, and you're just comparing them, you see, oh, this latter didn't change. That means this part has been methylated. And the good thing of this technology is it gives you, in theory, it gives you 1 base pair resolution of sequencing the methylation reading. So in the previous approach, in the previous one, you got a region, right? You use immune precipitation, it's still DNA fragments, and you probably know there is something happens in this DNA fragments, but you don't know exactly which nucleotide the methylation occurred. However, if you use the bisulfite treatment, that gives you 1 base pair resolution for where the methylation actually occurred. And the large-scale bisulfite DNA sequencing has been successfully conducted. I think it was also last year. I didn't look for the publication. I remember it was last year in Nature. and they did the whole genome bisulfite sequencing, I think, for one of the cancer cells. But there's really not too many studies published so far. And the major reason is, first of all, it's time and resource intensive, right? So if you really want to do this in the genome-wide scale, you have to, for any single individual, you have to sequence the original genome, which is a pretty big task, and you have to sequence the bisulfite converted genome. So you have to sequence 2 genomes at least to get this where the methylation occurred. And that is one problem. And the second problem, I think, is also major problem. It's about bioinformatics analysis. If you think it through, human genome are 4 letters, A, C, G, T. And we were talking about alignment and for almost an hour, and we still don't know how to do it, right? And this is a very daunting task. But after you do the bisulfite treatment, you are not doing with something that has 4 letters anymore. Because some of the C has been converted to U, some of the C are not, right? And when you do the alignment, how are you going to do it? It will be really, really, really hard. And it's not even 3-letter genome. It's a 3.5-letter genome, right? So you've got an A, C, G, T, and that's the 4 letters. But if it's complete conversion, you got a A, T, G. You don't have any C if all the C has been converted. But now it's not. Part of the C has been converted, part is not. How do you do alignment at that stage? It becomes really, really difficult. And I think that's one of the reasons, and this approach hasn't been used as often. Probably the first one, because you have to sequence both 2 genomes, that is more difficult, and because people are really concerned about the money. And the bisulfite sequencing and the low resolution or having focus in detail in the small genomic fraction, we'll talk about this in a moment. And the other problems is once you do the bisulfite treatment, you are hoping that all the unmethylated C has been changed, but sometimes you got an incomplete conversion, and that will interacting with your methylation signals. If some of them are not methylated but still not being converted, how you're going to do the inference, and that is some pretty difficult. And degradation of DNA during bisulfite treatment, that's another thing that people are pretty struggling on that as well. So what I'm talking about, all these problems or difficulties are for the whole genome bisulfite treatment. Okay? There are some other approach called the reduced library-based bisulfite treatment. And I haven't seen too many publications, but actually there are some pretty established protocols on that. So what happens is, I heard about Illumina talk about this once in one of their conferences. What they do is they treat the DNA with some restriction enzymes. So they know exactly where the restriction enzymes will cut. Okay? And then they run the gel of the fragmented DNA, and then some of the part is pretty big, others are pretty small, because of it purely depends on where those restriction enzyme sites are, right? And then you do the size selection. You just focus on small percentage of it, and then using those further do bisulfite treatment. Okay. Now, you're missing a lot of information. And but what they do is they will, before they do any experiment, they will systematically evaluate every single potential restriction enzyme. So they use this one to target. They will see where the cutting will occur. And if this part, they have some important things they want to focus on, they will pick this restriction enzyme size and do the size selection. And that sometimes can recover over 80% of the regions they are interested in. And there are some computational effort. I saw one paper in Nature methods a couple of years ago that they just systematically evaluating all the restriction enzyme and trying to tell you that which part, how to select those based on your potential goals. And I haven't really read that in much detail, but that is one way to do it. And there are some companies that you can send the samples in, and you tell them, here are the regions I'm really, really interested in. Please just focus on these regions. And then they evaluate all the restriction enzymes, and then they design the proper size of the gel where it cuts, and they do pretty good job on that. So the good thing of that type of approach is you get away from this whole genome situation, right? So if you think a lot of repetitive regions, they are heavily methylated. I'm not interested in those regions. And if you design the restriction enzyme right, and you should be able to get away from those regions. You can have better handling on only focusing on the regions you're interested in. All right? So have some collaborators at Purdue are sending their samples. Haven't seen the results yet, so I cannot speak of that. But it sounds like a very promising experiment to do. And so we talk about the immunoprecipitation-based assay. We talk about this bisulfite treatment, and this is the third type of approach, which is the methylation-sensitive restriction enzyme methods. So what happens is there are some restriction enzyme will only cut the CGs with has not been methylated. So those are if you use the enzyme to treat this, and you can see this part is unmethylated, it will be cut. If this is methylated, it will be protected, not being cut. And if this cutting really occurred, and then once you do the sequencing part, you will be able to just know exactly where the methylation occurred, because our sequencing can only happen from the beginning of the sequencing rate, right? So if you have a cutting at this location, and your sequencing will further along these lines. And then once you do the bioinformatics, you see a lot of signals in that particular region, you know that cutting has occurred. If there's another CPG site and the cutting were not detected, and you probably know that it has been methylated. So, and this is kind of it's called the methylation-sensitive restriction enzyme methods. And there are some enzymes that targets on unmethylated sites, and there are also some enzyme target on methylated sites. So you can choose the enzyme that you're interested in using and do this type of analysis. Is that okay? Is that understandable? There's a problem for this type of approach. Is most of the restriction enzymes, they are very much sequence specific, right? So it's not only determined by whether this part is methylated or not. It's also based on the flanking sequences as well. And so you end up with only measuring the methylation site on a subset of all the potential methylation sites, not all of them. So this is the potential problem of that. So for all this methylation, the sequencing-based methylation detection, and there's a pretty good review paper that you want to go into and take a look at that. Okay? And the last topic I want to talk about in terms of epigenetics is chromosome looping. So many people start to consider this is one of the epigenetic mechanisms. So you can see that although DNA molecule, they are kind of linear, but actually in the real cell, they may not be linear. So they may have this kind of looping type. So we know that that is one of the mechanisms how enhancer actually work, right? So enhancer, if you put on the DNA linear phase, it's pretty far away from the targeting part. But actually, if DNA loop in this way, and that it's very possible that this protein complex bring this looping back together, they are physically actually pretty close to each other. And the DNA looping allows the transcription factor bonds to a distant enhancer to interact with RNA polymerase or general transcription factors at the promoter regions. So exactly like that. But the issue is how do we really detect this type of DNA looping? Okay, and people becomes very smart. The first approach is there's a review paper published in Cell Research in 2009. And the first approach is called 3C. I don't remember what that 3C stands for. And but what happens is you got this one end, there's another end, and they're pretty far away from each other, but they're physically together like this. So what happens is for this one, you just cut this part, and then you want to ligate these two parts together, and they are becoming this. And then you do the pair end sequencing. So if one end that after mapping is in this location, the other end is in this location, and they are, you know that in the real cell, they are physically pretty close to each other. Okay? So what happens is you got, again, this is a protein, and they bring these two DNA molecules together. You do some cutting here, and using restriction enzyme sites, and then you ligate them back together, and then you sequence from both two ends. That's the way to do it. Or this is a more sophisticated approach, is you not only ligate them together, but add up some type of linkers or some kind of bait in this region, and eventually you can concentrate the signals. You only focus on these ligated sites. And this is called Hi-C. I think this is published in Science in 2009. And it's very similar to 3C. The only difference is you put some kind of a biotin labeling in this ligation sites, and then you have some ways that only concentrate your signals, only eventually focus on those ligation sites, and then to sequence from both two ends. We have some data for both 3C and Hi-C approaches, and this method sounds understandable if you read the papers, but once you do it, it's kind of really not that nice. So for one of the first sample that we got is for the 3C technology, and we found probably only less than 0.5% of the sequencing rates are usable, and the remaining are not. So we got a couple of Illumina runs, and that was pretty expensive. But 99.5% of the rates we detected, those two ends are very close to each other after mapping. Okay, so it really didn't capture this distal sites. And eventually, our collaborator changed the protocol to Hi-C, and that helped a lot. And only 97% of the rates are not usable. And we got to improve that from 0.5% of usable rates to 3%, which we are pretty happy about that, right? So these technologies, they seem pretty straightforward, I mean, conceptually, but once you really do that, there's a lot of hustle to go. And I think this is what I'm going to talk about today for the epigenetics part. Just a brief summary of what we really discussed. And the first story is RNA polymerase. And so you can see that using that information that we can study microRNA, or we can study other type of transcription regulation. And the second one is for the histone markers. And there's not too many methodologies that has been developed, but we do have one methodology that can be used that identify the enriched regions. It's not a peak finder, because histones doesn't work in that way. It's not a peak finder, but rather it's finding out the enriched regions. And the third topic that we talk about is information. So basically, that's the three different assays that regarding DNA methylation, which is the immunoprecipitation-based assay, or the restriction enzyme-based assay, or bisulfite sequencing-based assay. Okay, so those are the information I want to deliver, and we will have another break, maybe 10 minutes, and after that we will have the course review. So we have about 60-something slides, and the point of course review is I want to briefly touch base what we have talked about in this whole semester, and those are really the knowledge point I want you to understand. I will make sure that this will be an open book exam, so it's not open to internet, so you cannot really use the laptop or things like that. It's an open book, so feel free to print out your slides, or you use computer, that's fine, but just don't do the Google part. But I will make sure that you wouldn't find the answers in my slides. So still try to understand the slides, and that will make you in a better shape. All right, let's take a 10 minutes break, and we'll do the review part.